I think the science has never been better in the world. The discoveries are accelerating at a pace that's never been seen before. We've got so many new tools to fight disease. We've got so many new tools to do drug discovery that we never had before. And so the pace of biomedical innovation is unmatched from where it's been historically.
This is the Nucleate Podcast, the voice of the next generation of biotech leaders.
Welcome to the Nucleate Podcast. My name is Sam Kessel, and on this episode, I speak with John Maraganore, co-founder of Alnylam Therapeutics, a pioneer in siRNA drugs. He's also the inventor of bivalirudin, a key drug in anticoagulation used in procedures around the world. He has a resume that's too long to recite, but he's been an advisor to VCs such as Arch Ventures, Atlas Ventures, Blackstone Life Sciences, and RTW, among others. He's also active with many nonprofits, and we are very proud to have him on the board of directors of Nucleate. Since our original recording, John has become co-CEO of Coursera Health, and we're excited to see how his new company progresses. So I'm curious, how would you explain what you do to a 5-year-old?
I make medicines. That's simple.
That is simple. So I'd love to learn a bit about your background. I know you grew up to Greek immigrants in Chicago.
So I very lucky in my life to have been born of immigrant parents. I think they like many immigrants came to this country with very little and with the dream of making having a family, building security, building happiness. And so, there's something about immigrant parents that, really drive their children to make the most out of their situation because they didn't have very much when they came here. Mm-hmm. And it really was a blessing in many ways because they drive you very hard to work hard and be industrious and study hard and study long hours. And they certainly did that with me as well. My father was a physician, and so he really taught me the love of science that I have, the love of medicine that I have. And my mother was a nurse. She stopped her work as a nurse when she had kids, but she also was in the healthcare profession. So I had the benefit of two parents who were immigrants. They wanted me to work hard and study hard and so forth. And then they were in the healthcare profession, so they inspired me to stay in that profession as well.
So do you feel like it was inevitable for you to become a scientist based on your background? How did that all play out?
Well, actually, they didn't want me to be a scientist. Mm. They wanted me to be a doctor. Mm. And they wanted me, my father in particular, wanted me to be a physician and maybe as we grew up and got our medical degrees, open up a medical office with him. And I mean, that was his dream. Mm. And I crushed his dream a little bit when I got a PhD instead of an MD. In fact, I'll never forget, he was very worried about whether or not I'd be able to make a living as a PhD scientist as opposed to being a physician at the end. So that was not, by far was not the path of least resistance. It was a path that was not promoted actively. But the path of least resistance was the fact that I fell in love with science, and of course, the microscope and the chemistry set and the biology set that I had when I was growing up. Those all helped me, be passionate about science. At the end of the day, I just went down a science path, not a medicine path, in my academic training.
So what made you make that decision? It sounds like medicine was being pushed on you a bit. What made you choose science instead? And what was the moment that made you make that decision?
Yeah. So I and in fact, I was a pre-med at the University of Chicago, and I was thinking about going to medical school eventually and so forth. But while I was doing my undergrad work, I got involved in doing research in a lab. There was a research lab that was looking for a research assistant as a job, and I applied. I was able to start working in the lab, and I fell in love with research. Absolutely fell in love with it. Would go after class and do my research, and take notes of the findings. And I was working on snake venom, phospholipases as a area of interest, and on the enzymology and the structure and the function of these proteins. And I just loved it, and it was clear to me that what's beautiful about doing research is you're exploring something that in some ways nobody else in the world is exploring at the same time. And there's that feeling of being on the frontiers, being a pioneer, being someplace where nobody else is at that point in time. And I loved it. I really loved it. So I just got the bug and decided, I want to commit my life and my career to science and research and pursue a PhD as opposed to going down the MD path.
So you got hooked onto research. You had some early successes and interests that really drove that. So I want to talk about your career as a scientist more broadly. A lot of folks who know you as the founder of Alnylam. But at the beginning, you were pursuing a pure scientific path. You started out at Upjohn and Biogen, and then at Biogen, where you invented bivalirudin, also known as Angiomax, which is used in many procedures all over the world. I'd like to get to understand more about your experience, especially with inventing bivalirudin and being the first to invent that drug.
Yeah, I mean, when I went to the Upjohn company after getting my PhD, I really fell in love with being involved in industry research and being involved in the biotech industry, and that was in the early '80s, right? So or the mid '80s. This was a time in which biotech was just starting. You think about, Biogen and Genentech were only 5-6 years old at that point in time when I got started. But what I found when I went from, the University of Chicago to the Upjohn company, and I did a postdoc there at the time, was that industry research is so much more collaborative and so much more applied. And unlike academic research where you're focused on one very, very narrow subject and you're the expert in the world on that very, very narrow subject, when you're doing research in a biotech or pharma community, you're part of a team of people that are working on a problem. And I also really enjoyed the concept that the problem you were working on might ultimately lead to a medicine that can be used in hundreds, thousands, millions of people at the end of the day. So I love that aspect of scientific research within a company. And for many years through about the early 90s, I was really active as a scientist within these companies, both at Upjohn and Genetics and at Biogen. And I was able to actually be involved in inventing a molecule that went into the clinic and got all the way to the market, and that's bivalirudin, or Angiomax. The discovery of which was really quite fascinating. It was not a sanctioned project at Biogen. Biogen had a rule, they used to call it the 20% rule, where they allowed and endorsed their scientific team to spend up to 20% of their time exploring some interesting area of therapeutic application that wasn't necessarily a formal program at the company. And that's what I did with bivalirudin. I had an interest in thrombin as an enzyme, as a potential way of generating a direct thrombin inhibitor that could be used as an anticoagulant. I was obviously, from my research work and in my PhD thesis, I was always interested in venom proteins, and the leech saliva has a protein in it known as hirutin, which is an anticoagulant. And I was interested in finding a way to make that hirutin protein smaller so that it would be potentially less immunogenic, and did that work in my 20% time at Biogen, and ultimately led to this very exciting discovery around a highly potent thrombin inhibitor, where we filed IP And then we began to do the translational work. It then became a formal project at Biogen. People at Biogen thought this is pretty cool. We got a new drug candidate. And it became Biogen's most exciting program for a while. And then ultimately, beta interferon emerged as being a more promising opportunity. But Angiomax ultimately made it to the market and helped millions of people around the world, which is super cool.
And that must have been very rewarding to do something that's so impactful. I'm just picking up on the thread that this was a surprise, or at least Biogen wasn't planning on developing this drug. This kind of came up in your free time. Was it a coincidence that you developed this drug, or did you know you were going to develop something that looked like bivalirudin and its success from the beginning? Or was it a surprise to you in that process as well?
Well, it was very much a surprise because again, I was taking advantage of this sanctioned space of scientists being able to spend 20% of their time, and I would typically do this work in the evenings before I would go home, or on the weekend I would come by and start doing some of the work, and because it's not a sanctioned project, you'd have to get other people within the scientific team to work with you on this. It was a peptide, and so there was a peptide chemist at Biogen who I needed to basically partner with to ultimately make the peptides that we would evaluate. And the part of it that was a surprise was that after we discovered the initial peptide molecule, and this is around 1988 or 87, 88. We found that there was a piece of intellectual property out there from a different company, different lab, that would have blocked us from being able to develop that one peptide. And so the project almost stopped at that point in time because we felt, oh my God, there's a patent out there. We'll never be able to make a drug from this. But we came up with an idea about making a bivalent molecule, a molecule that would stretch from one portion of the thrombin structure to the active site of thrombin, and by being bivalent would have a higher affinity as an inhibitor. And that was the real aha moment that led to this sub-nanomolar potency thrombin inhibitor that ultimately went and had free intellectual property from an IP standpoint and ultimately got into the clinic. So it was a very accidental discovery. I remember doing the initial work again late one evening and looking at the kinetics of thrombin inhibition in a enzymatic assay and being blown away with how potent the molecule was. And we really discovered something very special at that time.
When you were at Biogen, you made a pivot away from the lab and into the business side of things, which is where you spent most of the rest of your career. Can you tell us a little bit about how you made that change and what was the cause of it?
I didn't make the pivot.
Okay.
The pivot was made for me. Okay. I was the head of the molecular biology department at Biogen, and then at the same time I was the project leader for this bivalirudin program at Biogen. And the project leadership role was really a super fun role. We can talk more about that if you'd like, but it's a role where you lead a multidisciplinary team that's involved in developing a product from preclinical all the way through phase 3. And we took the molecule all the way through phase 3 and came to a result which was not exactly what we expected. And so Biogen decided that they would no longer advance that program, that they would put it on hold and try to find a partner to basically take it forward. And we eventually found a partner for the molecule. But for me, that led to a change in my responsibilities. I no longer had a project to lead because we put it on the shelf. And around that time, the CEO of Biogen, a guy by the name of Jim Vincent, very notorious, that's probably the wrong word, a very well-known, highly respected, but a bit controversial leader in the biotech industry at the time. He was the CEO, and he called me into his office and he said, "John, you've done drug discovery, you've done drug development. I want you to do business development." And I said, "Oh, okay, that's good." And so I left his office, and I was really distraught. I did not want to leave science. and he was asking me to leave science. And so I sort of ignored him. He called me back in his office about a week later, and he said, John, I just wanted to check in. Have you thought about this transition from the science side into the business development side? I said, yeah, I have. I think I'm okay staying on the science side. And he started advocating for why I should make this change to business development. Now, after I left his office, I realized it wasn't being a choice I was offered. It was, you either do this or you don't have a job anymore. And I had recently gotten divorced. I had, child support payments to make. You had to split my income in a significant way. I couldn't not have a job at that point in time. So I agreed reluctantly to go ahead and make this change in my career path and lead up business development at Biogen. And initially, I was unhappy about that change. I didn't really want to do it. But within a few months, I realized this is a great opportunity. I was meeting lots of companies. I was helping find partnership opportunities for Biogen. Biogen had a strong balance sheet. It had the ability, the firepower that was needed to basically be able to do some exciting business development transactions. I did a few deals over that period of time, including partnering my baby, bivalirudin, with a company called the Medicines Company, which we did in 1997. And so it was really super fun to be involved not only in finding new opportunities for Biogen on the business side, but also being able to take your own invention and find a home for it at the end of the day. So super fun experience, but it wasn't my desire to go do it. It was forced on me. But of course, I am so thankful and grateful that I ultimately had that opportunity, because it gave me the business experience that I needed to then ultimately become a CEO, which I did 6 or 7 years later from that point.
So I'm guessing then that it wasn't your plan to become a CEO. That was also, in a way, thrusted on you in a similar fashion that how you got into the business development experience. You had all the capabilities you needed across the scientific side, clinical development, and then the business piece was really the linchpin at the end that was put together.
Yeah, but I have to say, I think after I got in the business world, becoming a CEO, of a company, starting, especially at the beginning of a company, did become something I was excited about doing. My original plan would have been to be a head of R&D, to be on the research side, and I admired people like Vicky Sato, who was the head of R&D at Biogen, and Richard Flavell, who at the time was at Biogen as well, obviously extraordinarily well-known scientist at Yale. But Richard was also on the R&D side at Biogen. That would have been my original plan prior to Jim Vincent forcing me into the business world. But once I got into the business world and I enjoyed that side of it, the potential of being a CEO was definitely on my radar at that point in time. And in fact, in 1997, when I left Biogen to go to Millennium, I was recruited there by Mark Levin, an amazing leader. And Mark brought me in to be the general manager, effectively the CEO of a subsidiary company within Millennium focused on biotherapeutic products. And I was excited to go do that with him, not only because of the opportunity to lead something, but frankly, the excitement at the time, again, this is 1997, around the Human Genome Project and how the human genome could transform medicine. And that got me super excited to go do that.
So that's a great segue to talk about your experience being a leader early on at Millennium. What was the original vision of the company, and how did that intersect with the work of the Human Genome Project?
Yeah. Well, I wouldn't call myself a co-founder of Millennium. I would call myself a co-founder of the subsidiary at Millennium. I was an early employee at Millennium for sure, and really had the wonderful experience of working with Mark Levin and Bob Tepper and many others that were there at the time. And it was an exciting time in science because everybody saw the potential power of understanding the human genome. And we all were excited about how that could transform how we do drug discovery, how we do drug development, how it can transform the whole future of medicine. And, I got that bug. Why wouldn't I? I'm a scientist at heart. And it really was something that was an exciting period. With the biotherapeutics subsidiary that we built at Millennium, we were competing against companies like Human Genome Sciences and a few others that were really focused on finding novel protein drugs out of the genome. The big mistake I think we made in hindsight is, we all thought that we can go from the structural information of the genome to predicting function of novel proteins. And as you guys know, most scientific work it goes the opposite direction. You have a functional activity and you isolate a structure at the end. And it turns out going from structure to function is a lot harder than, observing a phenotype or a functional activity and then isolating a structure at the end. But it was still a very successful effort. And during my time at Millennium, we ultimately brought the subsidiary back into the main company. I then led M&A at Biogen, or Millennium, sorry, where we acquired a company called Leukocyte at the time. That was done in 1999. And then in 2000, we acquired a company called Core Therapeutics, and really transformed Millennium from being a genomics discovery company into being a fully integrated biopharmaceutical company. And really amazing transformation of Millennium over those few years. And I was part of the team with Mark and Kevin Starr and Bob Tepper, who we're very close friends, but we worked together very hard to make that transition happen at the end. Steve Holtzman, another key person in that whole story as well.
So you ended up spending 5 years at Millennium, and then Phil Sharp called you and invited you to get involved in Alnylam with him. Can you tell us about what those early days were like at Alnylam and what allowed you to take that leap of faith from Millennium, which is still going relatively strong when you left? And this was at the peak of the genomics revolution hype, and you had a stable career pathway at Millennium while you were there.
Yeah, no, I mean, the story is super interesting. I mean, it actually started in 2001 when I get a phone call from Phil Sharp, who I knew from my Biogen days. And Phil calls me up and says, John, I thought I'm reaching out to tell you a little bit about RNA interference and some discoveries that I've made with some of my coworkers here at MIT, and we'd like to come by and talk to you about it. So, Phil came over with 3 other people. One is Tom Tuschl, the other is Phil Zamore, and the fourth person was Dave Bartel. And they came to Millennium and they presented the story around small interfering RNA molecules mediating targeted mRNA degradation in mammalian cells. And it was an amazing presentation. Tom Tuschl, presented basically his data out of his lab. And it was a stunning discovery because up until that point, RNA interference was viewed largely as being a phenomenon that was restricted to plants and maybe some primordial organisms like the worm. And the original work by Farah Mello, as you know, was done in C. elegans. So it was a big surprise to discover that RNAi could work in mammalian cells. And for Millennium, it could have been an opportunity to transform functional genomics because you can now systematically interrogate the function of a given RNA in a cell culture system. And so we got very, very excited about that. We ended up taking a license to the MIT technology for that purpose. But then about a year later, I get a phone call from Peter Barrett at Atlas Ventures. And Peter is somebody I've known for a long time, wonderful venture investor. He calls me up along with Jean-Francois Formella, who is also at Atlas Ventures. And they told me about Alnylam. And I had read that, the company had gotten started. I kept an interest in the RNA interference world after that meeting with Phil. But the call I got from Peter and Jean-Francois was really the first approach to me to go lead Alnylam, which at the time they had just put $2.5 million into the company. There were a couple of employees that they had hired at that point in time, and they needed a CEO to come in. At the time, Christoph Westphal, who's now at Longwood Partners, but at the time was at Polaris Ventures, was serving as sort of the acting, CEO as the venture person, but they needed a full-time real CEO to come in. And so they reached out to me, which was terrific. Now, I started reading up on the science, looking at, the issues with making drugs out of the RNAi pathway or that harness the RNAi pathway. I obviously realized that it was going to be super hard to do it. These molecules, small interfering RNA molecules, they're big, they're charged. They'd have to get into the cytoplasm. People were using transfection to get them into cells, but you couldn't just incubate a cell with these molecules and get them inside the cell. So we'd have to figure out ways of achieving delivery, and we knew that was going to be a big challenge. But I couldn't stop thinking about the potential, and I, over time, over the summer of 2002, over several months. Period of time, I would just keep reading about the science and kept thinking about the potential. And I made the decision in October of that year, 2002, to make the leap and go from Millennium to run Alnylam as the first founding CEO of the company. And it was an interesting time for me personally. I was getting remarried, and my wife Christine, who was super supportive of my thought process at the time, always reminds me to this day that my offer letter to join Alnylam was actually sent to my honeymoon suite in Mauritius after we got married, where I signed it and sent it back by a fax. But it's sort of a fun story. In many ways, we got married, of course, and had a family, but in other ways, I also got married to building Alnylam, which was a tall order and a fun order as well.
And that must have been one of the best days ever. I'm curious, you mentioned challenges with delivery as well as with immune response. How did you navigate those issues as CEO and make the decisions that you needed to make the platform successful?
Yeah. Look, I mean, first of all, we had eyes wide open on what had to get done. We knew that we'd have to figure out ways of achieving delivery, and even in the early days we thought about lipid nanoparticles. We thought about conjugate-based approaches. We thought about other polymers that might be used for delivery of these molecules. We also knew that we'd have to potentially stabilize these molecules in some way. Initially, the immune response aspect of it wasn't as well appreciated until a few years later, where it was clear that certain TLR receptors could be activated by siRNAs, as well as the RIG-I pathway could be activated by siRNAs. And so we had to figure out how to deal with that as well. And we ultimately figured out that chemical modification would attenuate any reactivity with the immune system. So we found, in stepwise fashion, just like science is typically conducted, we found all the answers to the many, many, many technical questions that existed at the time when we got started. And remarkably, we ultimately were able to figure out how to make drugs out of small interfering RNA molecules. But it took a long time. It took the better part of a decade before we really knew how to do it. It took probably about $1 billion of invested capital over that period of time to really figure out how to do it. It took a lot of hard work and trials and tribulations and false dead ends many dead ends that we went into, including, lots of things that we would do with other third parties. We took a very collaborative approach to try to find the answers. to what we're doing with other companies, with other academic labs, It was a huge effort, really was a huge effort, because we knew that we had to ultimately solve these technical challenges before we can make drugs and make medicines. And thankfully we did. But it was a really a remarkable endeavor, scientifically and speaking, to really get to the point where we can confidently make medicines out of small interfering RNAs.
So you said it took a long time to get Alnylam off the ground. And I think it requires a different investor profile than what we're seeing today. A special kind of investor profile to have that incredibly long view. In 2025, when you're looking in hindsight on Alnylam, it seems obvious. You all have multiple commercialized drugs that are making a huge impact. But at the time, you're still in the clinical stages, and there was a lot of uncertainty in the platform. And after 10 years, you didn't know that you were going to get a commercial product at the time, right? So how did you navigate that? The requirement for you mentioned at the time is north of 10 years. A lot of investors have 10-year fund cycles, so they're not able to have that much longer term view, and they want to get a return on their investment. How did you navigate that environment, that push and pull?
Well, it's interesting. I mean, first of all, we got started in 2002, as a reminder, which was after the tech bubble had burst. And it was a very risk-off period in biotechnology, just like it is today. In many ways, very, very similar. Maybe not as bad as today, for other reasons we can talk about, but it was certainly a tough environment back then. The venture investors that came into the company were Cardinal Partners, Polaris, Atlas Ventures, Arch Ventures, and then Abingworth came in as well. And they had the vision to see the potential of RNAi as a new class of medicines, even when the public markets were pretty, dismal and very, very uncertain, I would say. The other category of people that were hopeful about RNAi were pharma companies. And so, we had strong interest from Merck early on to do a deal. We did a deal with them in 2003. Then we had strong interest from Novartis to do a deal. We did one with them in 2005. And so pharma really saw through the uncertainties and figured out that this could get solved, and they bet on us to basically try to do it. And the combination of the pharma deals that we did and the broader enthusiasm that emerged around the RNAi field allowed us to go public pretty early. We went public in 2004 with only 2 years of operating time in the company. And that brought in a whole new set of investors. Now, our public offering was not a marquee public offering, I would say. We raised $32 million on a $98 million pre-money valuation, which is laughable in this in the more recent IPOs that have been done. But back then, it was not unusual to get that type of IPO done. But we became a public company, and we brought in a number of investors, and we just executed. We showed them the data that we could make drugs out of RNAi. We took a very transparent approach to our science. We published many papers, including in journals like Nature, on the progress we were making scientifically. We presented our data in peer review forums where we developed a reputation of being a very high quality science-based company, and investors took the view that these guys might figure out how to make drugs out of RNAi. And of course, then the other thing that happened is the pharma interest continued to grow. One of the best things that happened in our story was in 2006 when Merck, who were a little bit upset that we did a deal with Novartis in 2005 because it was a pretty big deal, they ended up doing an acquisition of Therapeutics, which was a competing company at the time, for $1.1 billion. That was a big number. And that strengthened investor excitement about Alnylam, in part because they thought maybe Alnylam would be the next one taken out at the end of the day, although that was never our intention. And that allowed us to then do a deal with Roche in 2007 where we raised $331 million. We then did a deal a year later with Takeda where we raised over nearly $200 million up front. I mean, these are big numbers for deals that were done at the time. Those were probably at the time the biggest deals of a preclinical type technology that had ever been done. So we were able to build a balance sheet, basically, that really buttressed the company's valuation. And so when 2010 came around and the world gave up on RNAi, we were actually in a good place. We had about $300 million of cash on our balance sheet at that time. And that allowed us to survive that dark winter for our science and technology.
What's interesting is that today, it's very difficult to be a preclinical public company. And you're seeing many fewer of those today. While there are some exceptions, I found it interesting that you were able to achieve a significant number of pharma deals that you're able to get as a preclinical company. And this is a strategy topic that's come up in the podcast a few times in terms of balancing developing out your platform versus handing off drugs to a pharma, where you're counting on another party to develop your drug. So I'm curious on how you navigated those partnerships. It sounds like the non-dilutive funding from these partnerships was very helpful to Alnylam. But how did you balance the strategy of developing your own products versus partnering some as well?
Well, we always wanted to develop our own products. From day one, my business partner at Alnylam at the time, Barry Green, who's an amazing colleague and business partner of mine. We worked together for nearly 20 years at Alnylam. He and I were always committed to making drugs out of RNAi that we would commercialize ourselves. We always had that vision from the beginning that we would take products to market ourselves. And the reason for that is there's actually no way you can build a multi-billion-dollar company without ultimately commercializing your own medicines. There's just no way. Now you can get acquired, along the way and maybe get to those type of valuations but you can't do it without ultimately getting to the market yourself. But we knew that we had a bigger than a breadbox opportunity of assets. And there was no way, if RNAi was going to be as expansive as we had hoped it would be, there was no way we could do everything ourselves. No way. And so we felt good about finding ways to partner with pharma companies, but retain sort of our core area of focus for ourselves. And we began over time to learn where the technology can be best applied, to basically have a good understanding of what were the type of products that we would keep for Alnylam and take to market on our own. And those turned out to be in the rare disease space, as you know. But in other therapeutic areas and other categories where you might need some extra muscle from a pharma company, made sense. Why not partner? It represented a relatively non-dilutive source of capital for the company. And to sort of illustrate it at its macro level, I mean, over the 20 years that I was there, we raised $7.5 billion of capital, and it's split 50-50 down the middle between capital raised from the equity markets and capital raised from pharma, remarkably. And so that source of the 3.25 billion that we raised, or 7.5 that we raised from pharma, allowed us to raise a lot less money from the capital markets and obviously maintain share price and avoid dilution as much as possible.
So you said the focus of Alnylam is, of course, rare disease. I was curious on how you landed on amyloidosis as the initial indication, or one of the initial indications that you selected. How did you come about that decision?
Yeah, in 2010, when the outside world gave up on RNAi and pharma started exiting the field, and people thought we were dead, but we hadn't lied down yet. We felt that we needed to make a transition. We were hit by a 2x4, and we felt that we had to make a more demonstrable transition from being sort of a broader platform company to really focusing on building a pipeline where we had high confidence that we could build pipeline assets. And we had a number of key criteria that we thought were really important as part of our R&D strategy. One was to target liver expressed genes, because we had delivery that we thought could work in the liver, and we needed to really focus on where technically we could succeed. The second part was we demanded human genetic validation of the targets that we pursued, because we felt that the way to asymptotically reduce our likelihood of failure was to really focus on human genetics and ground ourselves in human genetics for the targets that we would pursue. A third criteria was that we would have an early biomarker that we can measure in phase 1 that would give us confidence that our drug was acting appropriately and that we knew the pharmacology of our product. A lot of drugs fail when you don't get the right dose in later development. So being able to get dosing understood and also tell our investors that our drugs were working early was important. And with those type of criteria and a few others, TTR really rose to the top pretty quickly. Monogenic disease, gain of function pathology, autosomal dominant inheritance, clear disease burden, significant prevalence of the disease. I mean, if you look for the genetic form of the disease, about 50,000 patients worldwide. If you include the sporadic form of the disease, so-called ATTR, CM, or wild-type disease, it's hundreds of thousands. So a pretty significant commercial opportunity, enormous unmet need. Patients needed therapies. When we started the program, there were no drugs that were approved at the time. It became a very logical place to go. We can measure the knockdown of TTR in phase 1, right? Very, very early. The genetics were really clear. Having said all that, there were still unknowns. I mean, it wasn't certain that taking a pathogenic protein that people have for their lifetime and don't develop disease until they're in their 30s, 40s, and 50s, would you really impact that disease if you knocked it down when they're in their 30s, 40s, and 50s? Or did you have to start when they were born? Okay, as an example. So there were uncertainties, but we felt that it was worth a shot. And we did a lot of diligence on the indication. We got convinced that that was the right program to launch as our lead program as part of that new strategy starting in 2010. And it turned out to be a good bet, for sure.
And of course, for the patients who are getting the drug, are certainly benefiting from it.
Amazing. I mean, I got to tell you, I for many years until we had the randomized data, we had some very interesting open label data. And you always have to be careful with open label data. It could just be patients believing that they're getting better because they're receiving a drug and so forth. But we were seeing some pretty remarkable, based on compared to the natural history, we were seeing some pretty remarkable effects of the drug from patients. And the physicians that were treating these patients were commenting to us, hey, these patients are doing a lot better. And they would comment on that. But we always sort of kept it, calibrated. We didn't want to get our hopes too high. But when we saw the randomized data in 2017, it was stunning. I mean, the approach of silencing TTR in this disease has a profound therapeutic impact on patients, and that was amazingly gratifying at the end of the day.
And one thing you touched on is that throughout all of this, in 2010, you were dealing with the challenges of the market during the Great Recession and the negativity around RNA interference at the time. Now, you mentioned strategically, Alnylam was all set at the time. You had partnerships, you had money in the bank, so you could weather through those tough times. But how did you maintain morale, whether it was for yourself or for your team? And how did you navigate those more challenging times, which I'm sure a lot of entrepreneurs in our audience are relating to very significantly right now with the challenging fundraising environment?
Right, I'm sure they are, and it was yet another tough time. I mean, between 2002 to 2010, it was more of a bullish market. But when 2010, 2008, 2009, 10 came around, you had the Great Recession, right? You had the Lehman Brothers collapse, you had the capital markets blow up, and here we were, about 8 years into building this company, and our pharma partners are just running away from RNAi, and then the media starts telling the story, because it's a story, and that's what they do. They like to report stories that this once wonderful, exciting technology is not going to make it, basically, as evidenced by the fact that the pharma industry is leaving. So it's pretty demoralizing, I would say. We started trading under cash as a company, which is not uncommon, especially now.
Yep.
Many companies in that same position if they're public, and it's a pretty demoralizing feeling. But it's interesting. We were looking at our data, and we were seeing reasons to believe, and we felt that even though the outside world, wasn't seeing it the way we were seeing it, we felt that we were right. We really, really looked critically at our data, and we saw glimmers of hope that gave us the confidence to keep going on. We also, in early 2011, we developed a 5-year plan that is somewhat, well documented now, called Alnylam 5x15, and we told the outside world, hey, we know you don't really believe in RNAi anymore, but we do. And to show you that we're right, we're going to bring 5 drugs into the clinic by the end of 2015 and show you clinical data around those 5 programs in a way that will excite you about the potential. That's basically what we said at that time. And it turned out, in the outside world, it didn't really register for a few years, but on the inside, it was pretty much a positive injection of hope on the inside. We had to do a restructuring at the time. We had to let a good number of our colleagues go. We had to do another one in early 2012. So there were some hard things that we had to do to preserve our balance sheet and our cash at the end. But that original 5x15 moment in Alnylam was one of the most energized moments in a very, very adverse environment for the company, because we rallied around this focus, and we rallied around the potential to bring it all back and make medicines out of the technology. And it really galvanized our team to do some amazing things. We ended up, at the end of 2015, we ended up having 8 products that we brought into the clinic with clinical data on many of them. And it was a pretty remarkable period of time. And we had rebuilt faith in the company. We rebuilt our valuation into the billions of dollars at that time, trading well above cash. We just needed to provide the clinical data to really convince people, understandably. But we had to show them the clinical data that RNAi could be a drug at the end of the day. We were able to do that over that 5-year period.
And in some of the darkest of days, you ended up having to let around 50% of the workforce go at Alnylam. I'm assuming that was a very stressful and challenging time for you as the CEO. How did you manage that for yourself? How did you handle that burden? How did you keep up with the morale of the team who was left? Was it a speech? Was it something else?
Personally, these are moments where having, your significant other or a friend, whatever it is, and is just so critical. I mean, my wife was instrumental for me because she would encourage me and say, you can figure this out. You can do it. I mean, she was my cheerleader. And she was the only person who I could really talk to about my feelings and my concerns and my fears and so forth. Because you really can't talk that openly with your team without, creating anxiety. You can't really talk that openly to your board because you don't want to create too much anxiety. Of course, you talk openly to them, but you don't express your real fears to them. And so having my wife by my side during that time was really super important for sure. But I did have a closer group of people, leaders within the company that really partnered with me. And we talked about Barry Green earlier. The other person who was instrumental in that time was Akshay Vaishnaw, who's the head of R&D, was the head of R&D. He's recently left Alnylam. But the three of us, and a few other people too, but just keep it to the three of us, we were very, very close. And we would be able to really challenge each other and think about how to make this all work effectively and successfully. We would, appropriately identify what the risks were, but where the opportunities lied, and focus on those opportunities. We would talk about, the business or scientific moves or development moves that we should make, and understand, the risks associated with that, but again, also the opportunities. So, having that sort of inner circle was super important. I think in biotech companies in general, there's so many examples of smaller groups of leadership that need to work with each other. Len Schleifer and George Yancopoulos at Regeneron, John Martin and Norbert Bischofberger and John Mulligan at Gilead. There's so many interesting stories like that. And I do think the CEO needs to have their personal cheerleader, maybe at home, ideally at home, and then have that sort of inner team within a broader organization that they can really count on. Because you can't do this alone. It's a hard job. It's a hard job. And having that support system, and especially when you can get really, really good input and challenge and critique from people you trust and really, really trust.
So after multiple successful drug launches and immense patient impact, you took a step back from operating from Alnylam after 20 years, and now you're a venture partner at Arch Ventures, RTW, Atlas, and of course, you're on the board of directors for Nucleate. I'm curious, from a bird's eye view, what are the most compelling trends that you're looking at in biotech across all of these experiences that founders should be aware of?
Look, I think the science has never been better in the world. The discoveries are accelerating at a pace that's never been seen before. We've got so many new tools to fight disease. We've got so many new tools to do drug discovery that we never had before. And so the pace of biomedical innovation is unmatched from where it's been historically. And that creates an enormous opportunity for new innovation to be born, for new ideas to be funded and brought forward into potential products. And I'm super excited about where all that's going. Even in these very challenging moments, and they are really, really challenging, there's no doubt about it, I'm very optimistic about where this industry goes over time to ultimately help transform the future of medicine and transform the lives of patients. And that's what this is all about. It's all about making medicines that transform the life of a patient at the end. And I think as long as we keep that in front of us, we will be in a good place, no doubt.
So one of the themes we've touched on a few times is that with grant funding cuts and a more challenging financing environment that we're in, how would you advise founders or aspiring founders, researchers, and others that are looking to start companies? How would you advise them to navigate this environment we're in 2025?
Yeah. Well, look, I think one thing I would say is this is a time where founders should make sure that they are ready to take the plunge into starting a company. And ready, from a personal experience perspective. This is a time where, access to capital is going to be far more discriminating. It's going to be far more limited to to people that have a level of experience that can convince investors that they ought to be banked at the end of the day. So I do think people should seriously think about whether or not a period of time, maybe 5-10 years, working in a great biotech company like Alnylam, or working at another large successful biotech company could be a great learning experience for them. So I think getting that experience right now at a time like this is a good idea in many ways, especially when the markets are very restricted and there's not a lot of capital flowing around. You sort of want to be in a place where you can learn at this point in time. But I also think that there are opportunities for new company creation. Venture investors are still investing capital, not as much as they were in 2021 or 2020, for sure. That's probably a good thing. I mean, they were probably over-investing in those years and probably allowing too many things to get funded at the end of the day. But you're going to have a much better idea of what you're going to do, why you're going to be differentiated. The bar has only gotten higher for what has to get done. I mean, the climate, I think, will improve. We've been through a dark period of almost 3.5 years now. Most of that's been driven by the interest rate environment at the end, linked to inflation. I think those pressures are going to get better in the coming year. So I'm optimistic about that side of it. The political uncertainty is certainly notable in this environment and unfortunate in this environment. But that too will settle down. I mean, there's going to be midterm elections next year that could change the dynamic of the uncertainty to some extent and allow a more stable environment for starting companies. So I'm generally optimistic here about where things go. I mean, I think obviously it's bad right now. It's super bad right now. It's probably never been worse right now, but it's going to get better. And the companies that survive this period, or the companies that get started during this period and get funded during this period, may be the greatest companies ever, because the bar for them to get started and funded has never been higher. And there's a lot of reason to be optimistic about where those companies can go. So we'll see how that goes over time. But I predict the crop of 2025 will be a very exciting crop of future companies at the end.
We certainly hope so. Now, I want to switch things up a little bit and transition to a quick fire round, which is where we're going to give you questions and you just tell us the first thing that comes to your mind. First thing is, I notice you have an R ring. What was your sleep score this morning?
I believe it was 75. 75 was my sleep score, yeah. And I didn't get as much sleep as I would have liked, but it was around 75.
Next burning question is, would you rather have deep dish from your homeland of Chicago or New York slice since you're on the East Coast now?
Deep dish. Deep dish any day. I was at the University of Chicago 2 weeks ago, and we went to the Medici restaurant and had some nice deep dish pizza, and it was awesome.
What's the trend or fashion statement from the 80s that needs to come back?
Wow.
Good question.
Disco? Perms?
Yeah, maybe some disco, maybe some new wave music. That would be fun. Yeah.
What were you doing the last time you felt absolutely at peace?
Skiing.
What is the wisest thing you've heard from a child?
Probably from my daughter Lexi, who is such a, she doesn't like me saying this, but she's so zany sometimes, and she would just say some of the craziest things. We had a few years back. She's now much older. But a few years back, we were having fun with her, and the things she says are just insane. Yeah. So definitely from her.
What is the theme song of your life?
Probably. What's the name of the song? Oh God! Don't stop now. What is it? Don't. What's the name of the song? It's
Don't Stop Believing?
no, that's a good one. But it's another one. But it's same genre.
Okay.
Same genre.
Yes, I know what you're talking about. I can
Yeah.
play it, but I'm not going to try it.
Exactly. And I didn't forget the name. Right.
Yeah.
I think it was Tom Petty that sang it. Yeah.
Do you have any books that you recommend everyone in our audience reads?
I mean, look, I think that there are wonderful books about this industry. I do think Peter Kolchinsky's book around drug pricing is notable for people to read. If they're interested in this industry and they want to understand how to articulate the value proposition and the business of medicines and why they cost so much and how does it work from a societal perspective, I think that's a really good book.
That's The Great American Drug Deal, right?
That's correct.
Yep.
Yes, indeed.
So to finish off, any final words of wisdom for the founders in our audience who are excited to start a company, want to follow in your path, and make a difference for patients?
Yeah, I mean, I would just say, first of all, you follow your dreams and follow the science, and most importantly, focus on impacting the patient. But don't be in too much of a rush. Remember, I didn't start Alnylam as CEO until I had been in the industry for about 17, 18 years, getting experience at Biogen, getting experience at Millennium. And I think it's really important that young people get that experience because it's super important. Making medicines and building companies is incredibly difficult, incredibly challenging. And it's like launching a moon launch at the end. And you have to be experienced to do it. You have to know what you're doing. So get that experience as well. And also, I'd say be in person as well. That's my other piece of advice. The virtual thing, not necessarily the greatest way to learn at the end.
Absolutely.
Perfect. On that note, John, thank you so much for joining the Nucleate podcast.
Fantastic.
Thank you.
Great to chat. Awesome.
Thanks for listening to the Nucleate podcast. Be sure to subscribe if you'd like to keep up with future episodes. See you next time.